Alright, so before we jump into chapter 20, looking at microbial diseases of the nervous system and the eye, let's go ahead and finish up chapter 19, looking at skin diseases. So before we talked about diseases of the normal skin flora, so diseases that you get from the staph, the Propionibacterium acnes, and the other organisms that are normal inhabitants of your skin that can still cause disease under certain conditions. So let's talk about the non-normal skin flora diseases. So one of the most extreme examples of a non-normal skin flora organism causing a nasty skin disease is Streptococcus, which can cause necrotizing fasciitis. And in particular, we're talking about Streptococcus pyogenes here. Not all species of strep will cause this. So Streptococcus species are not normal inhabitants of the skin. Some can be normal inhabitants of your mouth and your respiratory tract, but they're not normally found on the skin. They actually, remember from lab, Streptococcus cannot grow in salty environments, cannot grow on an MSA plate, so strep species will not be able to grow on your skin normally. However, a Streptococcus can cause an infection of the skin, and then a good example of that is Streptococcus pyogenes, which does this by invading tissue. So it can start an infection on your skin, and then it can invade into your tissue in order to cause an infection and grow where it can grow better. So it can't grow on your skin, but it can grow deeper down inside your body. So how Streptococcus pyogenes invades tissue is through secreting a couple toxins. One toxin is known as exotoxin A, and because remember if it's exotoxin, we know it's being released by the cell, so it's being secreted out of the cell. Exotoxin A gets released by strep pyogenes into the blood, and what it does is it basically stimulates your immune system to overreact and cause a pretty strong immune response. And how that helps strep pyogenes, because you might think that's counterintuitive. Why is it creating an immune response? Isn't that just going to kill it off? What it actually does is your immune response is a little too much, so it actually is going to damage your tissue. Your immune response is going to actually kill human cells and further damage the tissue, which is going to allow the strep pyogenes to further invade and get further into the tissue. It also secretes a compound known as streptolysin S. So this is, you can tell by the streptolysin, this is kind of a strep-specific toxin. And what this compound does is it kills human cells, which again is going to allow the strep pyogenes to invade further into the tissue. And this invasion is very fast. So this destruction of tissue, which is actually going to be kind of eating of flesh, they call it flesh-eating bacteria. You may also hear flesh-eating virus. That is not what this is. In fact, you should know that a flesh-eating virus, that could be a virus because viruses don't eat. So there's no flesh-eating virus, but there is a flesh-eating bacteria, and that's strep pyogenes. So this destruction of tissue is very fast. It can be a couple of centimeters an hour can actually be destroyed by strep pyogenes. So this is, because of that, a medical emergency. One very characteristic of necrotizing fasciitis is that strep pyogenes help spread along the muscles. So that's how it moves along, is actually along your muscles. And what that does is it causes intense pain, because now you're getting death of muscles. That leads to a very intense pain. So this is considered a very painful condition. So one of the characteristics of this is having a red spot that's extremely painful to the touch, much more painful than it should seem for what the wound is. Unfortunately, this is a very unfortunate, nasty condition to have. Streptococcus and necrotizing fasciitis can lead to organ failure and death in about 50% of patients. How you get this is through either direct contact with someone who is infected with it, and I should say that if we have strep pyogenes, because we did use that in lab, we didn't use a pathogenic version, but let's say you were to spill strep pyogenes, even if it was pathogenic, onto your skin, that's not 100% that you're going to get necrotizing fasciitis. You have to have some sort of a wound already to allow this to occur. But either through direct contact or the parenteral route, so you could directly inject yourself with Streptococcus pyogenes, and this could cause this condition. So here's an example of a blister of someone who has necrotizing fasciitis. Very unfortunate and very disgusting to look at. The diagnosis for this is actually kind of difficult because the symptoms at first seem relatively non-specific. A lot of times an individual will have flu-like symptoms at the beginning of this, and they'll have some intense pain and swelling. But one of the things that does make characteristic that you should start to suspect necrotizing fasciitis is if the skin is really hot to the touch. So if you see pain and swelling and really kind of this redness that's really hot to the touch, and also if you see signs of dead tissue, and that's dead tissue. Whenever you have dead tissue, you're going to have a lot of pain. So that's another thing that's very characteristic of this. Having a very low blood pressure, so the exolysin A that this organism produces, the overactive immune response it causes, sends your body into a system of shock. And that's going to lead to a very low blood pressure, so that's very characteristic. And also having, like I said, pain that's not really equivalent to what the wound would suggest, so really intense pain. So how they treat this is basically removing the affected tissue, and depending on how long you let it last, that could be a little or it could be a lot. And then also treating with IV antibiotics. Fortunately, as of yet, I'm not aware of any Streptococcus pyogenes strains that cause this that are incredibly resistant to antibiotics. But usually when you treat with antibiotics for this, you do a combination of both clindamycin and penicillin. So why do you think you might want to do both antibiotics? I'll let you guys think about that, and we'll talk about that. Why do you think we want to treat with both of these? There are a couple of reasons why we treat with multiple antibiotics over just one. So let you guys think about that. So Pseudomonas is another organism that is not normally found on the skin but can cause an infection on the skin, and in particular we're talking about Pseudomonas aeruginosa. So Pseudomonas aeruginosa is a gram-negative aerobic rod, and you've actually used this in lab several times before. Pseudomonas was one of the organisms that we use in the oxidation fermentation tubes, and we saw that it is capable of oxidizing but not fermenting. Pseudomonas is well known for having an extremely versatile metabolism, so it can grow on a whole bunch of different compounds. We actually used Pseudomonas when we compared the organisms that could grow on minimal media versus those that could not, and how we supplemented it, and Pseudomonas was capable of growing on pretty much very minimal media as long as it had some sort of a carbon and a nitrogen source. So it can grow on a whole bunch of different compounds. One of the places where we find Pseudomonas most frequently is soil. Is a big one. Also, anytime you have decaying matter or any sort of a moist environment is a really good place for Pseudomonas to possibly be there. And because it has such a versatile metabolism, it can grow in a wide variety of places. One of the unfortunate places we see Pseudomonas in hospitals is infections on burn victims in particular. And so where we see Pseudomonas a lot is actually growing just under the surface of the burn. Because Pseudomonas is an aerobe or aerobic rod, it does need oxygen to grow, but if you have a burn victim that's got some exposed tissue, that's a really easy area for Pseudomonas to get in and start to cause an infection, growing just under the surface of the burn where it's still able to get some oxygen. Treatment for Pseudomonas is inherently difficult because Pseudomonas is kind of a jerk and can degrade many antibiotics. Not really a jerk, it's just defending itself, but Pseudomonas is very able to degrade antibiotics that target it. It does this through, usually when you're treating for Pseudomonas, you're going to use multiple antibiotics. And again, think about why you'd use multiple antibiotics. Polymyxin being one that's used as a last resort, because it's kind of a really nasty drug, one that we don't like to use too much. But sometimes we have to because Pseudomonas is so difficult to treat. It's actually been shown that Pseudomonas can live in solutions of antibiotics as long as there's some sort of a source for them to grow on. They can actually live. So it's a very hardy, difficult to treat organism. So how can you identify a Pseudomonas infection? So think about when we use Pseudomonas in lab, was there anything in particular about Pseudomonas on a plate that made it stand out that you could use in an infection? And the answer, yes, there is. So take a look. Pseudomonas is a green organism, so it grows in green. You see this green here growing under the surface, under the bandages of this poor individual, this burn victim. Pseudomonas is green, so that's how you could possibly tell Pseudomonas. It also tends to have a very fragrant smell, so I'm not going to say it smells good because a lot of you don't think that bacteria smell good at any point, but compared to some of the other organisms you smelled in lab, like E. coli, Pseudomonas actually does smell very floral. It has kind of a fruity scent. So what Pseudomonas does is it is really good at killing cells. And it does so because it secretes several toxins and virulence factors that help it to destroy tissue and then trigger shock in the individual. Kind of like Streptococcus pyogenes does, but obviously it's going to have its own kind of virulence factors. It doesn't secrete the same ones. So the virulence enzymes that Pseudomonas uses, one is exoenzyme S. And exoenzyme S is an enzyme that inhibits protein synthesis in eukaryotes. So basically it's going to prevent our eukaryotic cells from producing proteins. You prevent protein production, you're going to result in death of the cell. So that's how it kills our cells. One of the ways it can kill our cells is through this exoenzyme S. It also secretes an enzyme known as elastase, which is targeted directly to destroy immune proteins. So it's going to destroy the immune proteins that your immune system is trying to produce to fight off the infection. So it's also got a good defense. It's going to destroy your immune system so you cannot destroy it. And then pyocyanin is another one. Pyocyanin is actually the compound, the pigment that Pseudomonas produces that makes it appear green. So this pyocyanin pigment it produces, that's actually kind of a bad thing. Pyocyanin actually triggers reactive oxygen species formation to help it destroy tissue. So reactive oxygen species is one of the ways that your immune system actually kills bacterial cells or any invading pathogens, is by producing reactive oxygen species and actually killing the cells. So when a macrophage swallows a bacteria, it destroys it by actually producing reactive oxygen species and basically bombarding that bacteria with all those toxic reactive oxygen species we talked about. So superoxide, hydrogen peroxide, all those different anions that make oxygen so toxic. Hyoscyanin is going to be produced by Pseudomonas to destroy your tissue. So it's actually going to trigger reactive oxygen species to further destroy your tissue. When it destroys your tissue, then it allows itself to invade further. And the reason it wants to do this is because your tissue is awesome food source for Pseudomonas. So it can grow and grow and grow if it can break down more and more of your tissue to allow it to grow. So the last bacterial skin disease I want to talk about that's caused by a non-normal skin flora is gas gangrene, which is caused by Clostridium. And so Clostridium are gram-positive anaerobic rods. I mentioned them briefly before, but they are spore formers. They're kind of like the anaerobic version of Bacillus. So Bacillus are aerobic spore formers. Clostridium are anaerobic spore formers. And so Clostridium are toxin factories. The organism that causes gas gangrene can produce 11 different types of toxins, which is actually pretty insane when you think about it. And all 11 of these toxins are basically designed to either kill your cells or to allow Clostridium to establish a longer infection in your body. So none of them are good for you. So how Clostridium develops is after ischemia. And so ischemia is going to result anytime you have an interruption of blood supply from a particular area of the environment, of the body. And oftentimes this is going to come after a wound. So often a wound has occurred that has resulted in damage that has blocked off blood supply to a particular area of the body. So not having blood supply is not good, because having blood means you don't have oxygen, because the red blood cells in blood are what carry the oxygen to the various parts of the body. And if you don't have oxygen, you're going to have anaerobic conditions. And anaerobic conditions for a long period of time will eventually result in necrosis or tissue death. You're going to have death of that particular part of the tissue. Now, if you've had some sort of an ischemia and Clostridium spores become present in the area of the body that blood has been blocked off from, what you have is an anaerobic environment that is absolutely perfect for the germination of those Clostridium spores. So this is why, one of the reasons why, after a wound, you have to be very careful, make sure you clean everything well and get it treated as quickly as possible, because if there were Clostridium spores wherever, whatever caused the wound, and they were able to get into the body, they can then germinate in this area that's been blocked off from blood supply. And so how can you tell if you have an infection with this? Is a couple of things. One, intense pain. So again, we're having tissue death. Tissue death is not something that doesn't hurt. Tissue death is very painful. So you're going to have intense pain in the area. You're also going to have blackening of the area. That's a sign of necrosis and tissue death. So blackening of the area, and gas bubbles. The gas bubbles are actually coming from the Clostridium, which as it's growing anaerobically, it's producing hydrogen and carbon dioxide gas. So we're having gas bubbles produced. Sometimes these bubbles will actually break and ooze. So treatment of this is incredibly important to happen as quickly as possible. You have to treat this rapidly, because Clostridium in these great anaerobic conditions can double every 12 minutes, which is actually insane. So one cell will divide 12 minutes, and then 12 minutes later, the cells will divide again and again. That is incredibly fast. When we grow E. coli overnight in the lab, we can get, at best, in the really enriched media, a doubling time of about 20-25 minutes. This is half that, so it can double in half the amount of time as our overnight E. coli can, which grows very quickly. So that's insanely fast. So how do you treat this is removing the dead tissue as soon as possible, providing antibiotics to kill off the organism, because you want to prevent the organism from growing. But the organism is not the only problem. The problem is the organism has actually secreted a lot of toxins as well, so you also have to provide antitoxins. So antitoxins function kind of like antibodies. Antitoxins are kind of like antibodies, where they're going to bind to the toxin and prevent it from doing its action. So antitoxins will basically negate the activity. They'll inactivate the toxin in the body, so the toxin can't do what it had been doing before. So here's an example of gas gangrene, kind of disgusting. So you can see the darkening of the tissue, and also there's these bubbles forming here. So these bubbles are gas bubbles produced, the hydrogen and carbon dioxide produced by the Clostridium as it's growing in the tissue. All right, that's enough about bacteria. Let's talk about some viral skin diseases. So diseases, the one you probably think of first is going to be herpes. So herpes is what we think, if you're going to think of viral skin disease, you're going to think of herpes. Herpes are slow-spreading skin lesions, so lesions on the skin that spread relatively slowly. And they commonly are what's known as recurring. And the reason they recur is because the herpes virus is an example of a virus that goes latent. And so it does that by incorporating its genome into your genome, which we've already talked about. And as a result, you never really get rid of herpes. Even if you clear off part of the infection, you don't have any active lesions that you can see, you still contain the DNA of the herpes virus in your DNA. And this is because of the viral, after the primary infection, the virus remains latent inside you. And it can reactivate later in life. Sometimes this can be very frequently later in life. Maybe it's not that late, maybe it's just a little bit of time. Or it could be much longer in life, depending on the type of herpes virus. So in terms of herpes virus, there's a whole bunch of different varieties, but the ones that we actually give the name herpes to, we usually refer to either herpes virus 1 or herpes virus 2. And so herpes virus 1 is generally considered the above the belt herpes. This is the one that's going to cause mainly the cold sores, fever blisters, things that happen above the belt. So usually the face, mouth, and trunk area. So a lot of people have had cold sores. You're familiar with this herpes virus 1. Herpes virus 2 is considered the below the belt herpes, and that's generally the herpes virus that causes the genital herpes cases that you get from unsafe sex. So the ones that you see in the genitalia area. But there are, I should say, this is not a tried and true rule. There are examples of herpes virus 1 that can cause an infection below the belt, and likewise for herpes virus 2. But generally speaking, herpes virus 1 is above the belt and herpes virus 2 below the belt. Alright, so how do we diagnose herpes? So the skin lesions of herpes are very characteristic. Here's an example of a cold sore here. Maybe you're familiar with having that before. So they're very characteristic skin lesions. And what's causing this skin lesion is death of the cells and then inflammation by your immune system in response to cell death. So as the herpes virus divides in the cells, it's going to kill them off. And as a result, the cells are going to die. The immune system responds to that and creates inflammation. So what you're actually seeing is the signs of your immune system responding to inflammation. So it's actually your immune system that's causing this. It's not like you're getting a lot of herpes that produce this lesion. It's the immune system responding to the herpes that actually produces the lesion that you see as it tries to repair the dead cells. Drugs can help control the disease, but there is no cure for herpes. Like I said, herpes goes latent. You're never going to get rid of it. There is no cure. You can control it. You can prevent future outbreaks, but you're not going to get rid of it. It's not life-threatening in adults. Usually consider herpes as more of an annoyance than anything else, but it's not life-threatening. It can be life-threatening in infants, however. So infants that get infected with the herpes virus, it can be a big problem. In fact, depending on how the infant is infected, the mortality rate can be up to about 30% if it's just a skin infection. But if it gets deeper in and actually the nervous system is involved, it can have an 80% death rate in infants. And so how do infants get exposed to herpes could either be with the virus crossing the into the placenta during pregnancy, although this is not one of the ways it occurs frequently. It's also possible that more frequently that the mother will pass it on to the infant either through the actual process of birth, so if a mother's infected with herpes when the child is born, or possibly if a mother has a cold sore like this, an oral lesion, if it were to kiss the infant, it could also pass it on that way. Alright, so we talked about herpes, now let's talk about warts, which are not to be confused with herpes as they are caused by a different virus, but they are caused by a virus. So warts are whenever you have benign growths on the skin or possibly mucous membrane. So here's an example of warts on the foot, so plantar warts that many people have had before, or I'll hear some warts on the face. So any benign growth you have, it's not cancerous, it's just benign. It's annoying and nobody really likes to talk about having them, although most people will get them at some point in their life. So what's causing warts is the papillomavirus. So human papillomavirus being one, so HPV that we've heard about, causes warts. Most of these viruses are harmless. So most of the times you get these warts, they're completely harmless. The virus that caused them is harmless. It's annoying, but it's harmless. How you get this is through direct contact with someone else who is exposed to this, who has a papillomavirus infection, and papillomavirus is able to go from one to the other, so you can transmit it through direct contact with someone who has warts. So there's various ways of getting rid of warts. Some can be there's all sorts of topical medicines you can put on it to prevent getting warts or getting rid of warts. There's also ways you can freeze the warts off. A lot of ways of getting rid of warts. Pretty common. However, new warts will develop, and a lot of this again has to do with viral latency. So much like herpes, HPV can go latent after the initial infection and then reactivate. So you can get new warts to develop later on. It's difficult to prevent getting warts, really just having proper hygiene and trying to avoid people who have them possible. All right, let's talk about the chickenpox virus. So chickenpox is actually caused by the varicella zoster virus, or VZV, which is a member of the herpes family. So again, we don't think about chickenpox as being with herpes. It ends in pox, so we automatically think of things like smallpox, but it's not. Chickenpox is actually in the herpes family. And so chickenpox, how it presents is basically a rash on the back and the trunk. Here's a young man that is showing a very characteristic rash from the chickenpox virus. And normally, mostly this is going to occur in children. So chickenpox is often a disease of children. They get chickenpox. Generally speaking, in children, it's actually relatively mild. It's really annoying. It's just like crazy, but they get over it. In adults, however, if you don't get infected with chickenpox as a child and you get it later as an adult, it can be a very severe infection. So chickenpox is not a joke when it comes to adults. So chickenpox is something that has been, well, in the past considered a normal childhood illness. Now they actually are vaccinating against chickenpox, so I'm not sure how long that's going to be. In the future, it's probably going to be considered not a normal childhood illness. But one of the reasons why there was a push to vaccinate against chickenpox is to avoid individuals experiencing shingles later on in life. So shingles is when the chickenpox virus, and again, since it's a herpes virus, it can go latent. It goes latent, and then so what happens is you get shingles, which is a rash. It's basically reactivated chickenpox virus. So the chickenpox that has gone latent reactivates later on in life to form a rash, and specifically where the chickenpox reactivates is going to be the skin along an infected nerve. So that's where the herpes, this particular virus, is going to go latent. So it's going to reactivate later on along a nerve, and you're going to see basically a rash on a nerve and a very severe amount of pain. And so the older you get, the more your risk for shingles goes up. And like I said, immunization is available for this. They are immunizing against chickenpox, and I personally did not get immunized against it, but I know a lot of people are being immunized against it. And again, one of the reasons is to prevent either, so if you don't get infected at a young age, you definitely don't want to get it later on as an adult, and also you want to prevent against getting shingles later on in life. Alright, so that wraps up the skin diseases. I want to transition now and talk about some microbial diseases of the nervous system and the eyes. Alright, so let's talk about the central nervous system here. So the central nervous system, or CNS, is considered axenic, which means there is no normal microbiota. So basically the central nervous system is supposed to be completely free of microorganisms. It's one of the aseptic or sterile areas of the body where you're not going to have any microorganisms present. So the question then becomes, how do pathogens enter the central nervous system? So if you look here, our central nervous system is consistent of our brain and the spinal cord, and it is very well protected. So there is what's known as the CSF, or cerebral spinal fluid, that is present in the central nervous system, is kept away from the blood and the rest of the body. So we have what's known as the blood-brain barrier. So how do pathogens enter the central nervous system? Is either through breaks between the bone and the meninges. So here's our meninges here, and here's the bone. If we have breaks between those, so normally this is a very secure, no breaks, but if you have a break, some sort of an injury occurs and you hear breaks occur, that could lead to pathogens getting into the CNS that way. Also through medical procedures is a possibility. So if you have any medical procedures that are going to result in injections into the central nervous system, such as a spinal tap, you could also mistakenly inject or have pathogens injected into the CNS that way. And it's also possible that they'll travel on peripheral neurons to the central nervous system. So if somehow the peripheral neurons around the spinal cord are able to, they get infected, then pathogens could possibly transfer along those to get into the spinal cord and then travel up to the brain or through the central nervous system that way. So the only way that the central nervous system gets infected is through something penetrating the blood-brain barrier. So something's going to penetrate the blood-brain barrier, that's going to cause an infection in the central nervous system. Alright, so let's talk about bacteria and the nervous system. So when you do have an infection of bacteria getting into the nervous system, some of these bacteria will directly infect nerve cells. Examples of this are meningitis, and leprosy. So these are examples where nerve cells are directly being infected. Other times, bacteria in the nervous system, bacteria might actually grow elsewhere, and they'll release toxins that are going to affect the nervous system. So this botulism and tetanus are both the examples of this. So both these organisms, which are caused by the Clostridium genus, they are going to actually grow elsewhere, but they're going to secrete toxins that are neurotoxins that specifically target cells in the nervous system. Either way, the nervous system is resultant, so we're going to consider all of these when we talk about diseases of the nervous system. All right, so let's talk about bacterial meningitis first. So meningitis you've probably heard about. Meningitis is one of the things they like to try to vaccinate you against before you come to college, as meningitis is commonly a problem in college, and specifically dorm lifestyle living. So meningitis is basically an infection of the meninges. So a bacterial meningitis would be a bacterial infection of the meninges. There are other types of meningitis as well, viral meningitis, fungal meningitis. And so what happens is you have a bacteria that has infected the meninges. So meninges are that thin layer of tissue that is helping support and surround the brain area. So one of the characteristics of meningitis is going to be a very sudden high fever and what's known as meningeal inflammation. So because you have an infection in the meninges, you're having inflammation from all the immune cells in the area trying to fight off the infection. And so this meningeal inflammation leads to encephalitis. And so encephalitis is when the brain becomes infected. When this happens, that's going to lead to changes in behavior. So anytime you're having infection in the brain, you're going to mess up kind of how your brain responds to environment, and basically you're going to have huge behavioral changes. Could possibly lead to coma and also possibly death. So meningitis is kind of a big deal. It's a big problem. It's definitely considered a medical emergency. So there are 5 species of organisms that can cause meningitis or have been shown to cause meningitis. So the most common one we think about is Neisseria meningitidis, and you can tell it's most common because we've actually named it after the infection itself. So Neisseria meningitidis is a gram-negative cocci, so it's actually the one we see here. We commonly see it as a diplococci, where you have 2 cocci together. Gram-negative cocci are relatively rare, so it's very rare to have this this particular type of gram-stain reagent. We don't have a lot of gram-negative cocci, Neisseria being the characteristic organism that does have this. So we have gram-negative cocci Neisseria. Also, Streptococcus pneumoniae can cause meningitis. So Streptococcus is a gram-positive cocci. Haemophilus influenza, it does not cause influenza, although it was named after that because it's thought that it did. It was thought to be the cause, but it's not. As we know, influenza is caused by a virus. Haemophilus influenza is a gram-negative rod. And also Listeria monocytogenes, which will cause actually a foodborne type of meningitis, is a gram-positive rod. And then last but not least, we have Streptococcus agalactiae, which is also a gram-positive cocci. Now, Strep agalactiae, I should say, is only known really to cause meningitis in newborns. And how newborns get infected with this is actually during the birth process. So about a third of women are natural carriers for Streptococcus agalactiae in their vaginal canal. And if a baby is born to a mother who is a carrier, they might pick this up and be born with bacterial meningitis. Not to worry, if you are a carrier, they actually do test for this, and they will, in order to prevent this from occurring and prevent your newborn from becoming infected, they will actually treat you with penicillin at birth to prevent the infection from occurring. Alright, so one of the things I want to talk about as far as Listeria, which is actually pretty cool, is the organism Listeria, that is normally a foodborne organism, how it evades your immune system. And like I said, just with meningitis, Listeria can actually cause meningitis after a foodborne illness. So what Listeria does is, so here we have a Listeria organism, is it allows itself to be phagocytized. So Listeria is going to allow your immune system to engulf it in order to kill it off. And when your immune system does that, so here we have Listeria being phagocytized into the cell, the Listeria is actually able to escape the vesicle that brings it into the cell. So here it is escaping from the vesicle. And then in order to move safely from one cell to another, so after this Listeria is going to replicate itself, and once it's replicated to enough, it's going to move on to another cell. Listeria actually takes advantage. It's able to take the actin in your cell, so use your cell's cytoskeleton as a way to slingshot it or kind of move it from one cell to another. So it basically takes the actin, bundles it together, and then uses that to spring it, so to basically kind of throw it like a rocket into a newly infected cell. So it goes directly from one cell to the other without having to go into the extracellular environment where all the cells that are trying to kill it off are. So it can evade your immune system by, once it gets engulfed, never having to be exposed to the extracellular environment again. So here's an example on an electron micrograph. Here's a listeria that's gone from one cell to another, and you can see the hole it's kind of made as it's directly gone from one cell to the other. It hasn't gone to the outside extracellular environment, and it's created this what's known as a pseudopod that moves it from one cell directly into another. So it's pretty clever what it's done, what it's able to do with your cell, to take advantage of the structure of your cell to move it, physically move it from your cell into another one of your cells. All right, so how do humans become infected with meningitis in the first place? So we've heard about it. It's not really good. Obviously it's a medical emergency, and it's a big problem in college communities. So one of the ways that infection occurs is through respiratory droplets. So it can be passed on, and this is why it can become, you get outbreaks of meningitis. It's because you get one person that gets infected with it, and they can transfer it on by respiratory droplets. Also during birth, this is the Streptococcus agalactiae example in newborns. Listeria can be transmitted by contaminated food. So listeria is not going to be transmitted by respiratory droplets. It's going to be by eating food contaminated with this really nasty foodborne pathogen, listeria. So how do we diagnose meningitis? Well, we have an infection that's in the cerebral spinal fluid in the central nervous system. So we have to see whether or not we have to do basically culturing from the cerebral spinal fluid. So the symptoms of meningitis are going to be fairly characteristic, and also if you suspect it, they're going to culture from the cerebral spinal fluid. So they're actually going to take a sample of your CSF and see if it contains organisms, and also does it have an elevated white blood cell count. And so the way they're going to do that culturing is going to be through a spinal tap in order to get access to the cerebral spinal fluid. Once they've done that, if they do diagnose you as having meningitis, they'll treat with IV antibiotics. There are vaccines available, so you very well may have gotten one before you came to college. So there are vaccines available against the Streptococcus pneumoniae, H. influenzae, and the Neisseria meningitidis version. So these are the three that if you do get bacterial meningitis that you don't get from contaminated food, and you're not an infant, likely you're going to be infected with one of these three. So there are vaccines available. Next, let's talk about leprosy. So leprosy, just like meningitis, is going to be caused by an organism that is directly infecting the nerve cells. So leprosy is caused by the organism Mycobacterium leprae. So this organism is in the so it's a mycobacterium. So mycobacterium, you should think of mycolic acid, because these organisms are going to have and are so named because they have mycolic acid in their cell wall. So it's going to be classified as a gram-positive rod, although it won't stain very well for that because of the mycolic acid in the cell wall. And so we talked about the mycolic acid. What that helps this organism do is it prevents it from drying out, so it prevents desiccation. It's also going to help it in terms of phagocytosis. So it's going to prevent this organism from being ingested by one of your phagocytes and being eliminated from the body. So it's really important in order for this organism to cause an infection that it has this mycolic acid in its cell wall. It's going to be very protective for it. So M. leprae has a relatively slow growth rate compared to other organisms. And the reason for that is that its optimal growth temperature is actually not at 35 degrees, like a lot of the organisms in your body, a lot of your mesophiles that grow on you. Its optimal growth temperature is actually 30 degrees. So it's going to grow best in the cooler parts of your body, which is going to be areas like your nose and the outer limbs, so like your fingers, your hands, your toes, feet. That's going to tend to grow better in those particular environments. And so there's a couple types. There's two types. There's tuberculoid leprosy, which is known as the non-progressive form of leprosy. And then there's lepromatous leprosy, which is also known as Hansen's disease, which is where you do get progressive destruction of the tissue, where the organism is actually spreading and causing tissue destruction to occur. All right, so leprosy is going to be spread from person to person contact, so basically what's going to happen is you get spreading from one person to another, and how it's going to enter the body, how it's going to get in, and eventually to your nervous system is through breaks in the skin. And so it's going to come in through breaks in the skin, and then it's going to travel along peripheral nerves to eventually get to the central nervous system in order to cause the infection. So how do you diagnose? Well, by symptoms. You're going to have symptoms where you have some sort of, you've all heard leprosy is kind of where you get some sort of death of the cells in the body, which is going to lead to basically physical deformations. We're going to see some sort of a physical deformity from occurring. And so that's going to be a pretty obvious symptom right there. Not a lot of things are going to cause physical deformities. And also by culturing from the area where you see these deformities are from the central nervous system and detecting for the presence of M. leprae. So if you can detect Mycobacterium leprae and you have these symptoms, that's a pretty good indication that the individual is suffering from leprosy. And so unfortunately you can't do a gram stain for this. Like I mentioned, the M. leprae having the mycolic acid in the cell wall doesn't allow it to actually stain very well, so you have to do what's known as an acid-fast stain for this. This is a very special way of actually testing. So acid-fast stain is used to stain for organisms that are mycolic acid. So if you do an acid-fast stain, you should be able to detect the M. leprae that way. So how do we treat leprosy? So they're going to use multiple antibiotics to treat for leprosy. The reason why they use multiple antibiotics is because this organism, M. leprae, can actually become resistant to just using one antibiotic pretty quickly, so that's why we use multiple. And they also get some protection against leprosy with what's known as the BCG vaccine. So this BCG vaccine was actually developed to target tuberculosis, but it's actually believed to work well in leprosy as well. Some protection, not great, but some protection from getting leprosy, because the organism that causes tuberculosis is also a mycobacterium organism. So they are related, and therefore some proteins they produce that the vaccines are going to be targeting are also related. So like I said, this is designed for tuberculosis, but also was believed to work well with mycobacterium. How do you prevent getting leprosy is basically limiting your exposure to the pathogen. So don't get involved with individuals that have leprosy or come into close contact with them. Or if you do happen to decide to go work in a leper colony and you want to help out, make sure that you take prophylactic antibiotics, which are basically preventative antibiotics, so that you don't get infected with leprosy. Alright, so because M. leprae prefers to grow in cooler areas of the body, this makes it kind of difficult to grow in culture. So think about why the M. leprae might be more difficult to grow in culture compared to other organisms. All right, let's talk of now moving from organisms that actually directly infect cells of the nervous system to organisms that start an infection elsewhere and then secrete toxins that directly target the cells of the nervous system. So let's talk about Clostridium botulinum and botulism, which you've heard of botulism. It's pretty well known. So botulism is considered to be an intoxication, which is not what you think it is. Basically, an intoxication is when you have an infection that is caused by a toxin alone. And so basically, you don't actually have to be infected with the C. botulinum cells. All you need to do is if you get infected with the botulism toxin, that alone can cause a problem. That alone can cause a disease. So intoxication is when the toxin alone can cause a disease in the body. So the botulism toxin is actually released by C. botulinum. And there are really three forms of botulism that can occur. So there's foodborne botulism, which would be an example of an intoxication, where you have only the toxin being released into the body. You have infant botulism, which is obviously going to infect infants. So this is when infants get infected with botulism. And then wound botulism, where you have an infection of C. botulinum that's gotten into a wound and then released a toxin that way. So the foodborne botulism is probably the one that's most well known, is the foodborne. So you've heard of, if you have cans and you see sort of a kind of a raise, swelling or inflammation of the can, so to speak, that would be an example of a can that might be infected with the organism that causes botulism. So what are the symptoms of botulism? So one, you're going to have a lot of weakness, very dizziness, vision's blurred, so a lot of neurological symptoms here. Also things like dry mouth, constipation, nausea, vomiting, so some neurological symptoms and also some digestive systems here. And the one that really triggers the head, that should at least trigger alarms in the body, in your head that you might have a botulism, is when you get progressive paralysis of voluntary muscles. So you're getting paralysis. So this is actually the toxin is affecting your nerves, and you're getting paralysis of muscles. So involuntary muscles that you could normally move voluntarily, now we have paralysis where you can't move them. So you get paralysis of the muscle. And so this is how the botulism toxin is actually causing the muscle paralysis. So here's an example of nerve cells. So nerve cells that release, that are come in contact with the muscle cell. So if we look at the nerve cells here that are going to release chemicals or neurotransmitters that are going to result in contraction of the muscle cells. Here we kind of blow this up and we see that we have vesicles in our nerve cells that contain the chemical known as acetylcholine. So acetylcholine is going to be released by our nerve cells, and it's going to make contact with the cytoplasmic membrane of the muscle cell. And when acetylcholine does that, that's going to lead to contraction of the muscle. And so this is known as a neuromuscular junction, where we're having a meeting of our nerves and our muscle cells. And so this contraction of the muscle is going to lead to movement. So this allows you to voluntarily move your muscles is when the nerve cells release acetylcholine into the synaptic cleft. So what botulism does is botulism basically prevents the nerve cells from releasing acetylcholine. So botulism is going to basically block acetylcholine from being released. And it's not going to do that at just one nerve cell, it's going to do that at all of your nerve cells. So you're going to block the release of acetylcholine, which means your muscles can't contract. And what that's going to do is it's going to prevent muscle contraction, which is going to lead to the progressive paralysis of voluntary muscles that we see when an individual is infected with botulism. So the botulism toxin is one of the most lethal substances that we know about, incredibly lethal with this. It basically has 1 nanogram per kilogram toxicity. So if you take the number of kilograms you have, it's not going to take much of this toxin to actually kill you. If you had 4 kilograms of the toxin and spread that out over, evenly distributed over the world, it would be enough to kill the entire human population. So it's really nasty stuff. Now, cosmetic Botox, in case you're wondering, cosmetic Botox works because it has less than 100 units in an injection. So an injection of Botox, if you ever plan on getting it, is going to have less than 100 units. 3,000 units is considered lethal. So you can see there's a big difference between the amount of units that are actually going to kill you and the amount that you're getting. That being said, it's always important to make sure that your healthcare workers or whoever is injecting Botox in you, I don't know who actually does that, but whoever's doing that, make sure they know how to do math so that you do not get an injection of more toxin than you should have. And like I said, the action of the toxin is to block the release of acetylcholine and therefore prevent your muscles from contracting, leading to paralysis. Now, how this works with your facial muscles is that it basically blocks what happens when you get wrinkles that Botox is used to treat is that you've actually had too much contraction has led to weakening of the facial muscles. And so if you prevent the contraction from occurring, it basically prevents the weakening or the kind of slack, basically wrinkles result when you get weak facial muscles. So if you prevent the contraction, you prevent the wrinkles from occurring, and it just tightens everything back up. So infant botulism is the most common form of botulism in the United States. And we get about 80 cases of it per year, which while it isn't a lot, it probably is 80 cases too many. Most of these cases are completely preventable. One of the biggest reasons what causes infants to get botulism is they ingest spores. So if you give an infant some food that contains spores of botulism, those spores will get into the infant body. They'll start to germinate once they get into the gut of the infant. Because the infant does not have the microbiota in its gut built up to prevent the spores from germinating, those spores are going to germinate. They're going to release the botulism toxin. That toxin is going to then travel into the nervous system of the infant and cause the botulism. This is why it's important not to give food that contains these spores to infants. The biggest culprit is definitely honey. Honey seems very mild. It seems like something infants would be able to eat, but it is not. Honey does contain spores of C. botulinum. Not a problem in our body because our microbiota, our gut microflora, is developed enough to prevent the spores from germinating. Infants don't have that. So if infants ingest spores, they can germinate and cause botulinum. So don't give honey to infants. How we diagnose botulism is based mainly on the symptoms, the neurological, and also the big one being the paralysis of muscles. Infant botulism has its own type of symptom known as floppy baby syndrome, which sounds funny. It could sound funny, but it's definitely not. And basically this is where your infant's muscles are so paralyzed that actually the baby becomes what's known as floppy baby. It can't hold itself up, and you get this kind of floppy baby syndrome. So treatment for botulism results in washing of the intestinal tract. So you want to completely flush out, if it's foodborne infant botulism, you want to flush out the intestinal tract. and treating with the human antitoxin intravenously. This is actually really important. So the antitoxin for botulism is the BIG4, and that's going to be given intravenously in order to basically bind to all the toxin in the body and eliminate it. So it prevents the toxin from interacting with the nerve cells to prevent the release of acetylcholine. To prevent that from happening, nerve cells can then stimulate muscle cells to contract, and the body can go on existing. Also, treating with antimicrobial drugs, you want to make sure you treat the actual infection, get rid of the organism that's secreting the toxin so that it cannot secrete any more toxin. And as far as preventing is basically destroy endospores in food. So all this again is tied mainly at the infant or foodborne botulism, but you want to destroy endospores in food in order to prevent this from occurring. All right, so botulism is on one side. Let's talk about another kind of related disease that results when a toxin released by a cell elsewhere in the body affects nervous cells. So another release of a neurotoxin, and that's going to be with tetanus. So tetanus is caused by Clostridium tetani, so another genus or species of the Clostridium genus. C. tetani produces a neurotoxin known as tetanospasmin. And so tetanospasmin has a slightly different mechanism of action than botulism does. And basically what tetanospasmin results in is muscle spasms or muscle contractions. So in tetanus, your muscles can't stop contracting. Kind of the opposite of botulism where they become paralyzed. In tetanus, they spasm and contract too much. And so what happens with tetanus, how you get tetanus, is having some sort of a break in the skin or mucous membrane that allows the organism, Clostridium tetani, to get into the cells, to get into the body, and then start to grow and produce the tetanus toxin. Usually it's not going to be the cells themselves that get in the body. Usually it's the spores of Clostridium that enter the body, start to germinate into the full organism that then releases the tetanospasmin toxin. And so the C. tetani will germinate in any form of anaerobic tissue. So it's important if you get some sort of a break in the mucous membrane and then you put a Band-Aid on and you basically cover it up, it gets anaerobic, that's where you might get germination of Clostridium tetani. So it likes anaerobic tissue. One of the big things we think about with tetanus is if you step on a rusty nail, that's basically going to be injecting, if there's any spores on that, directly into your body through the parenchymal route. It's going to be injecting directly into an anaerobic environment that then will allow this organism to grow and produce the toxin. And this is going to, again, release of the tetanospasmin toxin. All right, so here's how the tetanospasmin toxin works. So if we look at our motor neurons here, so here's our motor neuron in the cell. There's basically for every motor neuron that's going to, so our motor neuron is eventually going to make contact with our muscle cell. Our motor neuron is going to have a stimulating neuron that's going to tell the motor neuron, yes, release, let's stimulate and release contraction. Let's release acetylcholine so we can produce contraction. And then the inhibitory neuron is going to tell the motor neuron, okay, that's enough, stop. So the motor neuron that does tell the muscles to contract is itself controlled by a stimulating and an inhibitory neuron. Both of these are important. You have to have something that starts the muscle contraction, but you don't want the contraction to go too long. You have to have something that eventually will stop the muscle contraction. So what tetanus does, the tetanospasmin toxin, is it basically binds to the inhibitory neuron and it's going to prevent the release of the inhibitory neurotransmitter that's going to counteract the effect of the stimulating neuron. So for every yin, there's a yang. So we have the inhibitory neuron that balances out the stimulating neuron. Tetanospasmin prevents that from occurring so that now we don't have anything preventing our stimulatory neuron. So this is good. Our stimulatory neuron is going to keep releasing its stimulatory neurotransmitter, and that's going to keep resulting in the release of acetylcholine. So we're going to get constant muscle contraction, unstoppable muscle contraction, and that's going to lead to the spasms and the excessive contraction that we see with tetanus. All right, so if tetanus is untreated, it's actually very deadly. 50% mortality if not treated. How you diagnose it is by the excessive muscle contraction or that really rigid. If you saw that child in the beginning, so when I talked about tetanus there, had that very kind of rigid, we call it lockjaw like structure where basically it's excessively contracted, muscles can't stop contracting. That's a very good sign of tetanus. Also, if you've had any sort of a wound where you know you might have actually picked up tetanus or come in contact with anything that would suggest you might have become infected with tetanus, associated with the muscle contraction would be a good key indicator that you might have tetanus. How do we treat it? Basically, you're going to clean the wound first off. You're going to clean the wound, get rid of, if you have any other organism there, clean it out so that any potential organisms that or tetanus that might have gotten in, you can try to clean it out. Injection with an antitoxin. So that antitoxin has to bind to the toxin to prevent it from preventing the inhibitory neurons from interacting. So you want to rebalance out the balance between stimulatory and inhibitory neurotransmitters with your neurons. And also, again, treating with antimicrobials to get rid of any more of the organism that's producing the toxin. And tetanus, because it's so dangerous if it's untreated and because it's really, I won't say easy to get, but if you do come into contact or have some sort of a wound, tetanus is definitely not a hard thing to become infected with. We do immunize against it. So the DPT vaccine that you might have gotten, or DTP vaccine, the T of that stands for tetanus. So there is immunization against tetanus. We do have a vaccine that is designed to prevent you, even if you do get exposed to tetanus, from coming down with a severe infection of tetanus. So we do prevent it by vaccination. All right, let's talk about viral diseases of the nervous system. So we talked about bacteria. Let's move on and talk about viruses. So viruses cause diseases of the central nervous system much more frequently than do bacteria. And so thinking of the structure of the central nervous system, think about why viruses might cause disease more frequently than bacteria. So just think about that. So what viruses can cause is meningitis, so an infection of the meninges, just like bacteria can cause. Anything you can do, I can do better. Viruses can also cause infection of the meninges. Polio is actually a viral disease of the nervous system. We don't think about it that way, but it is. And rabies is another good example of a viral disease of the nervous system. All right, so meningitis. This is caused mainly by a group of viruses known as the enterovirus. And so how viral meningitis is spread, it's basically person to person by respiratory droplets or possibly by fecal contamination as well. So the enterovirus, shouldn't be a surprise to you, is normally going to infect the enteric cells. So it is a cell that normally can be found in fecal matter. And so what the enterovirus does is it actually attacks cells in the intestinal tract naturally. So that's where it's normally going to infect. So how do cells get from the intestinal tract elsewhere? It's going to be spread by the blood. So enterovirus might get into your intestinal tract, cause an infection, kill some cells in the intestinal tract, and because it killed cells, it's able to spread from your GI tract into the blood. And from there, it's going to be spread to other areas. So at this point, if you have a virus in the blood, you have what's known as viremia, and it can be spread to all other areas, including, of course, if you have a break in the meninges of the brain or some sort of a break in the blood-brain barrier, this virus can then be spread from the blood to the meninges, where it can cause an infection of viral meningitis. So the symptoms of viral meningitis are similar to bacterial meningitis, but they're just a lot more mild. And in general, when it comes to this, your immune system is going to tend to respond to a bacterial infection a little more strongly than a viral infection, just based on the fact that it's triggered by bacterial much more easily. So think about how you might go about diagnosing viral meningitis if you were to do this. So how would you diagnose this compared to bacterial meningitis? So how they typically diagnose viral meningitis is by having typical meningitis signs and symptoms. So you got all the signs of meningitis, but you don't have any bacteria in your cerebral spinal fluid. So looks like you got meningitis, it looks like meningitis, it smells like meningitis, but we don't see bacteria. Generally, that's going to be a hint that this is a case of viral meningitis. Any treatment for viral meningitis? Nope. And the reason why is because it's a virus. Viruses in general, when it comes to treatment, just doesn't really happen. So the best thing you can do with viral meningitis is work to alleviate the symptoms. So basically make yourself comfortable and hope that your immune system will win the fight, which usually it will. Usually it will win the fight. Prevention of viral meningitis: wash your hands. Make sure you avoid things like crowded swimming pools. Don't breathe in respiratory droplets of someone else who has meningitis. Basically, take good care of proper hygiene, and you should be able to avoid viral meningitis. All right, let's talk about polio. So poliomyelitis is caused by the polio virus. And so we've heard of polio. We don't have a big problem with that in this particular country, but it is a problem in some underdeveloped countries, although we are getting closer and closer to eradicating polio from the world. But because it's been such a problem in the past, you should be familiar with polio and how it's caused. So polio virus is going to be transmitted by the fecal-oral route. So basically that's where it comes from fecal and it goes to oral. So somehow you're getting through the oral route, you're getting fecal matter contamination. That sounds gross, but it's actually easier than you think to happen. So signs and symptoms of polio. Generally speaking, it's going to cause an asymptomatic infection about 90% of the time. So basically, no signs and symptoms most of the time. However, there are some times, so minor polio, which causes about 5% of cases, is when you have very non-specific symptoms. This would qualify as the kind of flu-like symptoms. I just feel sick, I just don't know what's causing it. So that's about 5% of the time. 2% of the time, you get what's known as non-paralytic polio, where you have some muscle spasms and back pain. Keep in mind, again, this is a neurovirus, so it's going to target cells of the nervous system. Muscle spasms and back pain, that's going to happen about 2% of the time. And then less than 2% of cases are going to be paralytic polio, which actually results in paralysis. And so those examples here, you might have heard of things like the iron lung related to polio, where you actually have individuals that are experiencing paralysis, and they actually have to be put into these giant devices that will actually breathe for them because they are not able to breathe themselves. All right, so this is a map showing the incidence of polio from 2002 to 2009. And so you can see that overall there have been things that have gotten a lot better. You can see a lot of countries are actually having 0 what we call wild cases or cases of polio. And some are even considered certified as being polio-free. But if you look, there are some areas. So India in particular is believed to be, has in the past been kind of the epicenter of polio. There's been a lot of problems with polio. However, India has actually been polio-free for a full 3 years now and actually is considering itself completely free of polio. Now, some of the paralysis that comes from polio is considered lifelong. Not all of it is, but some of it is. So this is why it's one of the a big problem. And a lot of times, even if you don't have paralysis forever after being infected with polio, you will get what's known as post-polio syndrome, where you'll have a deterioration in function of muscles that were affected by polio. So when the virus got into cells and started replicating and infecting and basically killing cells of the muscle and nervous system, is you actually will have a deterioration in function where those muscles won't work as well as they did before you got polio. So even if you're not paralyzed, you are going to show effects usually for a long time after, if not forever. And again, as of January, India has gone now 3 years without a polio case, which is actually a pretty big deal for India. So that's really good achievement for them. All right, so the last virus that causes a nervous system disease is rabies. And so rabies is going to be transmitted, this is an example of a zoonotic infection, so it's going to be transmitted by bite or scratch from an animal that is infected with rabies. So a lot of different animals can be infected with rabies. We often think of things like raccoons, possibly dogs, that's kind of rare, squirrels, a lot of wild animals could be infected with rabies, like possums. You might get organisms that do have rabies, and so if you come into contact with them, get bitten or scratched by them, you can get rabies transmitted to you pretty easily that way. So what rabies does is it's going to replicate in the muscle cells and going to move on from there to the neurons. So the virus starts in muscles, then moves on to the neurons. And so what you get with rabies, the initial signs and symptoms is going to be some sort of a pain at the area of the bite. Of course, the bite or scratch itself is going to hurt. But you're going to get pain, a lot of itching, the classic flu-like symptoms, and also anorexia. So we're getting some neurological kind of anorexia. You're not hungry and you just don't eat. Later signs and symptoms as the disease progresses is going to be a lot of neurological symptoms here. You're going to have a fear of water, things like seizures, hallucinations, and eventually also paralysis. And so what happens, why this disease is fatal, is actually you're going to have a death from the respiratory paralysis. So if you get a paralysis of your respiratory muscles, because think of your lungs as being like muscles, paralysis of that is going to result in death. All right, so how do we diagnose rabies is going to be, well, these neurological symptoms are pretty characteristic of rabies, and also a history of animal association. Generally speaking, you're going to be able to tell if you've been bitten or scratched by a wild animal or an animal that might have rabies. Those are the kind of thing that usually doesn't escape your attention. So having that history and also combining with the neurological symptoms that will appear is a good way to diagnose rabies. They can also do antibody tests in the blood, so they can test for whether you contain antibodies for rabies in your blood. Since the rabies virus is not normally part of your body, if you have the antibodies for rabies virus, that means that your body at some point was exposed to rabies, in which case you are likely infected with rabies. The only reason you would be producing antibodies to it is if you had been infected with it. They also can detect for Negri bodies in the brain. And so these Negri bodies here, those are these kind of dark bodies you see here. These are cells of the brain. Those are actually formed. That's one of the characteristics of an individual who has a rabies infection in the brain, is they're going to have these Negri bodies present in the cells of the brain. And so these can be tested. You can also test to see if the animal was infected. So if you got bitten by an animal and you happen to have the animal around, they can actually do a test from the brain cells of the animal and see if the animal was infected with rabies before they start you on the course of treatment for rabies. So how you treat with rabies is with injections, which are known as the HRIG. HRIG stands for human rabies immunoglobulin injection. So basically being treated with antibodies to the rabies virus. So this is actually, because the rabies virus actually divides relatively slowly in the body, this is actually the same as a vaccine. So you get treated with, basically it's like a rabies vaccine. You get vaccinated with it after you've gotten it. So, but because the rabies virus divides slowly, your immune system is able to build up an immune response, recognize rabies, and then target the rabies that's already in you. So because it goes so slowly, your immune system has time to get this done. So as far as prevention for rabies, basically control rabies in domestic animals and do your best not to come into contact with wild animals that might care for rabies. Rabies is very controllable. It's not something that's just rampant all over. You can avoid getting it. And the nice thing about rabies is you can usually tell when you do have it. But you should still be aware of it because it does happen frequently enough that we do need to be aware of it in the healthcare field.